So as I had said earlier that the DNA is self-complementary and the DNA allows the two strands of a parental DNA molecule to separate and to act as the template on which the daughter strand is synthesized. Now, this process is called replication, and the main enzyme which brings about the polymerization process is DNA polymerase. Now, even though the replication of the simplest DNA seems to be simple, but it is one of the complex multi-step process involving several enzymes. It's literally one of the complex multi-step process. So if we look into the objectives of today's class, we'll be going over the leading strand and the lagging strand, and we'll be doing the different replication proteins, different enzymes that will be playing a huge role in how the replication process is coordinated, how DNA replication occurs. So we start with the enzymes, DNA helicase, single-strand binding proteins, topoisomerases, RNAs. So primase is coming first. Primase, DNA polymerase, sliding beta clams, RNase H, and DNA ligase. So as you can see, we are having a set of enzymes of which you have only learned about polymerase and its structure in the past class. So we'll be going over all these enzymes one by one and to see what role they play in the process of DNA replication. So now coming to the first of the enzymes, that is helicase. As you can see, the DNA helicase is actually catalyzing the separation of the two strands of DNA, as you can see. Now these enzymes, as you saw, this enzyme binds to and moves in one direction along the single strand. So it has a directionality. Along the single strand using the energy of the nucleoside. I'm just replaying it. It is using the energy of the nucleoside triphosphate to displace the DNA strand. So it's using, you can see it's using the energy of the nucleoside triphosphate. Now DNA helicase acts as at replication forks. So you can see this is the replication fork. It is a hexameric protein. It's made up of 6 subunits and it assumes the shape of a ring. As you can see, the ring-shaped protein complex encircles one of the two strands at the replication fork near the single-stranded, double-stranded junction. So if this is the replication fork, this is the single-stranded DNA and this is the double-stranded DNA. It is encircling one of the single-stranded DNA at the single-strand, double-stranded junction. Just like polymerase, DNA helicase can act processively. If you remember the property of processivity, which was also showcased by polymerase, that is, each time helicase associates with the substrate, once more let's see the replay, each time it is associating with the substrate, they unwind multiple pairs of DNA. And one more thing, as you saw, the helicase are moving in a defined direction. So either they move from the 5' to 3' end, or they move from the 3' to 5' end. So there is unidirectionality to the enzyme, and this property of helicase is called its polarity. So helicase is a processive enzyme and a polar enzyme. So that is DNA helicase. Next, we move to the single-strand binding proteins. The moment helicase separates the two strands, as you can see, these proteins bind to the single-stranded DNA so that the DNA do not again form a double-stranded complex. There's one thing that is to be noted in case of single-strand binding protein is that binding of one molecule to the DNA strand promotes the binding of another SSB immediately to the adjacent single-stranded DNA. This property is called as cooperative binding. So the moment one molecule binds, it facilitates the binding of another molecule of SSB to it. And as a result, very fast, the whole single-stranded of DNA becomes surrounded by the single-strand binding protein. The property by which one SSB promotes the binding of another SSB is known as cooperativity. So SSB is an enzyme which exhibits the property of cooperativity. Now, next, I'm moving on. Oh, sorry. Let's start this one. Next, we are coming to the enzyme topoisomerases. Now, the thing is that the moment DNA helicase starts moving along, opening the double-stranded DNA, what happens? Supercoils become introduced in front of the helicase in this manner. Because DNA is basically a double helical molecule. The moment you select a point and start opening up the DNA double helix, automatically the twist number in the double helix increases, or the double helix in front of the replication fork becomes it becomes twisted. So, or supercoils becomes introduced like this. Now, if there are supercoils in the DNA, how will the helicase move forward? So, there are enzymes known as topoisomerases. What they do? These topoisomerases, in fact, help in removing the supercoils that are found in the unreplicated DNA part, double-stranded part in front of the replication fork. Once the supercoils are removed, then the helicase can move forward without any hindrance. The mechanism of action of these topoisomerases are that they do this either by breaking both the strand or by breaking one strand of the DNA without letting go of the other DNA and passing that single strand through the DNA so that the coils are relaxed. We now move on to the next enzyme, that is primase. I talked about primase the other day. It is a specialized RNA polymerase that produces a small RNA primer of roughly 5 to 10 nucleotide long. The primase identifies the 3' end of the DNA. As I said, you just should always remember the 5' end of the primer is bound to the 3' end of the DNA. So the primase identifies the 3' end of the DNA and accordingly synthesizes a primer of around 5 to 10 nucleotides long. And this primer is very essential because the last year we studied that this primer actually provides the 3' O group or hydroxyl group so that the oxygen can make a nucleophilic attack on the incoming alpha beta bond of the incoming DNTP. So that is primase. We learned about primase the other day. Now coming to polymerases, as you can see, so we understood other day how polymerase functions. So this is a very simplified diagram where you can see there is a 3' end and polymerase is extending the 3' end. This is a 3' end found here also. The polymerase will also extend the 3' end this side. But you saw that there is this concept of leading strand and lagging strand now coming up. Why is this a problem? Because if this is the replication fork and if the direction of replication is this way, so the replication is moving in this, the fork is moving in this direction. Now the fork is moving in this direction. Look at the direction of the primer. This primer has a 3' end, and we know only that the 3' end can be extended. So this primer will be extended along this direction. Hence, this particular red color new daughter will be synthesized in the direction of the replication fork. So no problem. The helicase is constantly opening and the newly daughter's DNA is being synthesized in the direction of the replication fork. So this strand is synthesized continuously and is also known as the leading strand. But if I look on the other side, you should have by now realized the direction of the primer. So let's go over this once more. If you realize the direction of the primer, this is the 3' end. Now the 3' end is in a facing exactly the opposite direction of that of the replication fork. Now, since this 3' end is facing an opposite direction of that of the replication fork, so you can see this strand is not synthesized continuously. It's broken and it's synthesized. It takes time. This works a bit faster than this one. Hence, this is the lagging strand. This lagging strand is discontinuously synthesized. So this is how we get the concept of the leading strand and the lagging strand. Just always keep in mind that in case of DNA replication, it is only the 3' end that will be extended of the primer. So the orientation of the primer and the recognition of the 3' end of the polymerase becomes very vital. So we know, I talked about another important point the last day, the processivity property of DNA polymerase. So you can see per second DNA polymerase binds to DNA and it moves on continuously and can add up 1,000 nucleotides per second. So this is the basic property of DNA. But this rate is not enough. So we need to speed this up. For speeding up this rate of processivity, I said the other day, we have a molecule known as beta sliding clamps, or simply sliding clamps, which bind to the DNA polymerase and increases its processivity. It drastically increases the processivity of DNA polymerase from just DNA polymerase alone can just add around 50,000 base pairs. And when the sliding clamp gets attached to the DNA, the processivity is dramatically increased to 5 lakh per minute. So this is one property we discussed the other day, that from 50,000 base pairs per binding event, to increasing the processivity to 5 lakh base pairs per binding event is facilitated by this molecule, which is known as the beta sliding clamps. Okay. Now, we always have to remember the DNA is, the new strand is synthesized on the mother from the 3' end. So the overall direction of replication is constant. It is the 3' end that is extended. So one is the leading one and the other one is the lagging one. Now, the short fragments of the DNA formed on the lagging strands are Okazaki fragments. And after they are named after the discoverer of the Okazaki fragments, Reiji Okazaki. He actually explained the phenomena or the formation of and the phenomena was there. He explained it and it was named after his honor. And they are usually between 100 to 1,000 base pairs in length. So these are short DNA fragments which are synthesized on the lagging strand, and they range from 100 to 1,000 base pairs in length. Now coming to the final enzyme, RNase H. Now, as you can see, H, if you look at the name of the enzyme, the H stands for hybrid. That is, RNase means any enzyme that can digest ribonucleotides. In contrast, DNase means any nucleotide that can digest deoxyribonucleotides. So that's the basic difference. RNase H means any enzyme that can recognize an RNA which is complexed with a DNA. So let's look here. This is the green color is the RNA, and this RNA is bonded to a DNA molecule. So it's a RNA DNA hybrid, and hence the name H. So these RNAs is actually capable of identifying those RNA molecules which are bonded to DNA molecules in a hybrid pattern. So this enzyme, as you can see, we have RNA primers. For the leading strand, there was only one RNA primer. For the lagging strand, we have multiple RNA primers at intermittent gaps. So to complete the process of replication, once the daughters have been synthesized, the leading strand being continuously synthesized and the lagging strand being discontinuously synthesized. Now what happens in between, we are having these RNA primers. Now these RNA molecules, the daughter should be completely having the DNA molecules only. So what is needed? We need to remove the RNA primers. How do we do it? So as I said, this is the uniqueness of a molecular machinery. There's a particular endonuclease, I mean to say ribonuclease in the name of H, which recognizes the RNA complex with the DNA. And this goes and specifically digests the RNA molecules. The moment the RNA molecules are digested, you will be seeing that there will be gaps automatically. In the lagging strand, we will be now having gaps between the Okazaki fragments because the RNase has digested the RNA primers. So what happens? So as you see, the RNase is digesting the RNA primers. And now as you can see, there are gaps in between these Okazaki fragments. These gaps needs to be filled. So here comes another DNA polymerase. The earlier class, I told you that there are 3 DNA polymerases, Pol 1, Pol 2, and Pol 3. So here comes Pol 1. The removal of RNA primer is leaving gaps. So DNA polymerase, what it does, it refills the gap. You just let me show you this thing once more. As you can see, there are gaps. Remember, this is the 3' end of the Okazaki fragment. This is the 5' end of the Okazaki fragment because the primer was here, present here. So if a primer was present here, if this was the 5' end of the primer, this was the 3' end of the primer, then this is the 5' end of the Okazaki fragment, and this is the 3' end of the Okazaki fragment. Note that the polymerase binds to this Okazaki fragment and will increase and will synthesize the DNA till a single nucleotide gap is left. See, it binds to this 3' end. So it's binding to this 3' end and is synthesizing it till a single nucleotide gap is left. Now polymerase is unable to seal these gaps. So another enzyme is required who will seal these gaps. Now, this gap is sealed by the enzyme which is known as DNA ligase. Here comes DNA ligase. It uses a high energy ATP to create a phosphodiester bond between an adjacent 5' O phosphate and 3' OH. So you see ligase is coming, it's using ATP and it is sealing the gap between the 5' phosphate and the 3' OH. Now, so we have done the first part, that is the basic enzymes required for DNA replication. Now, as we saw in that particular animation which I showed you, that the two strands are in two different directions. One of the strand is synthesized in the direction of the replication fork. The other is being discontinuously synthesized against the direction of the replication fork. And you saw that the orientation of the polymerase enzyme is also different. For one strand, it was facing the replication fork. And for the other strand, it is facing away from the replication fork. Now, the point is that when the two DNA strands are opened up, they need to be synthesized together. Now, in the animation for simplicity, it was shown that one enzyme is synthesizing the leading strand and the other enzyme is synthesizing the lagging strand. But that is the case, but the two enzymes do not move by their free will. They are attached or tethered together at the replication fork. So how does this phenomena happens? So the thing is that for the DNA polymerase to act, both the DNA polymerase, who are now present in two different directions, they are facing two different directions, to act at one point, the whole process should be coordinated very stringently. And for that reason, the two polymerases are held together in a protein complex. It's a multi-protein complex. And the last day, I had told you the structure of this multi-protein complex. We have the tau protein, the delta prime, the gamma loader, and these are the core polymerases, alpha epsilon theta, okay? Now this is the core polymerase and the total structure is held together in the via the gamma loader complex to form the holoenzyme. It is to be noted here that it is the holoenzyme, I mean to say the gamma loader complex. This is the gamma loader complex and this is the tau protein. The tau protein is holding the two polymerases to the gamma loader complex. Why is this known as the clamp loader or the gamma loader complex? Because initially the sliding clamp protein binds here and then it is loaded to the polymerase here, or it is loaded to the polymerase here. So initially the sliding clamp binds here and it is loaded to the polymerase either here or to the polymerase either here. Hence it is known as clamp loader. It helps in loading the clamp. And the two polymerases are held together by the tau protein so that the polymerases do not roam away in their free will. They are coordinated, they're kept together at the replication fork where one DNA is being synthesized in one direction and the other DNA is being synthesized exactly in the opposite direction. Now, how does this happen? So we'll be moving on to the Tombrone model of DNA replication. Just concentrate and look into the animation first for 2 seconds. See how the machinery is working. If we look into this particular structure, you see this is helicase. Through the helicase, a single-stranded DNA is being pulled. The ash color strands are the mother strands and the red color strands are the daughter strands. Okay, just a second. The red color strands will be the daughter strands. This is the replication fork. And as you can see, the helicase has wound itself or encircled one DNA strand and is moving towards this direction. If this is the 5' end of the mother, this will be the 3' end of the mother. And on the 3' end of the mother, the primase has recognized the 3' end of the mother and is now synthesizing a small primer 5' to 3' end of the mother. Sorry, so this is the 3' end of it. So the polymerase will be synthesizing a small primer on this 3' end. So we will be having a 5', 3' primer. This primer, let me proceed a bit more. See, if you look into the direction of the primer, this primer is having a 3' end on this side, which is the direction of the replication fork. On the other hand, we also saw that primase synthesized a replication fork where it recognized this as the 3' end and it started synthesizing a replication fork exactly in the opposite direction. Okay. Now, this is the polymerase holoenzyme which has sat on the replication fork. But prior to the sitting of the polymerase holoenzyme, primase and helicase needs to be present at the replication fork. In fact, helicase sits first, followed by primase, and then polymerase comes and sits. We'll go into that order later on. Let us understand the molecular machinery that is working here. So, we now have the two primers oriented in opposite direction. Now look at the loader clamp, the clamp loader complex. You see the sliding beta clamp has been loaded here. So the clamp is first loaded here. You see the clamp is in an open form, a ring, it's open. This ring will now encircle the polymerase of the leading strand. And when the polymerase of the lagging strand will start synthesizing, it will also encircle a second one. So this is recycled. A second clamp will encircle the lagging strand. There is one more thing you should note. They are present at the same position. The polymerase of the lagging strand and the polymerase of the leading strand are attached to the loader complex. So the polymerase cannot move. But what can change? The loader complex can increase its distance. I mean to say, you see the tau subunits, they are parallel to each other. But the tau subunits, if required, can move to this much also. So it can open up. So there is a flexibility of the tau subunit. The tau subunit is like this now. The tau subunit, if requires, can move to this side. So it's flexible. It can move flexibly along this hinge. Now, when the process starts, let's start. You see, the leading strand synthesis has started already, but the lagging strand has not yet started. And you see through the helicase, the single strand DNA is being pulled. You see it's being pulled. The primer direction is this side. So the single stranded DNA is being pulled. Let's move to some more point. And you can see the position of the polymerase is same. The single stranded DNA is now being synthesized through the DNA polymerase in the lagging strand in a discontinuous manner. Why discontinuous? Because this primer has already been synthesized and this portion of the DNA has increased. Daughter has been synthesized. But again, you see the mother DNA has been pulled out. So we need to have a fresh primer here so that this DNA can be synthesized. So you see, this portion of the DNA has been synthesized, but this portion of the DNA mother has been pulled out. Now this strand needs to be synthesized. So for the polymerase and the sliding clamp to leave this, they have to leave this portion now, and they need to bind to that primer. Let's see. See, the sliding clamp is being picked and the DNA is being synthesized. Just one thing to be noted, when the sliding clamp is being picked, see, when the sliding clamp is being picked, see once more, when the sliding clamp is being picked, the leading strand is halting for few seconds. For the polymerase, see, check once more, it's being picked, it's halting. The polymerase sets in and then the leading strand again synthesizes. See, the lagging strand is being, sliding clamp is being picked by the lagging strand. Let's do it once more. The RNA primer, the sliding clamp being picked by the lagging strand, leading strand halted, and polymerase is starting synthesizing. So this is the point where the leading and the lagging strand, and hence it is known as lagging strand, because lagging strand is going a bit behind of the leading strand. And to keep in mind that when the lagging strand primer is capturing the sliding clamp and is bringing it to the polymerase for synthesis further, at that moment there's a pause in the synthesis of the leading strand. This model of DNA replication is known as the Tombrone model. And according to this model, as you can see, the single-stranded DNA template of the lagging strand is being pulled through the DNA polymerase. It's being pulled, you see, it's being pulled through the DNA polymerase. Similar to the slide of a Tombrone, this flexibility allows the DNA polymerase to add nucleotides to the 3' end of the growing lagging strand. Now, there are other things also to be noted. The moment one Okazaki fragment is being formed, you see, now concentrate on this part of the thing. This is one Okazaki, this is another. RNase H removing the primer, Pol 1 synthesizing, ligase fixing it. Once more. RNase H removing the primer, Pol 1 filling the gap, ligase sealing the gap. Again, RNase H removing the primer, Pol 1 filling the gap, ligase sealing the gap. So this is a complex machinery in which, as you can see, a lot of enzymes, polymerase, holoenzyme, sliding clamp, helicase, RNase H, Pol 1 and ligase are acting in tandem to make this process of replication go on smoothly.